Okay, so good morning, everyone. And first of all, I'd like to thank the organizers for inviting me to this place. And I would be extremely happy to interact with you as far as I can, knowing my field. So let me introduce me a bit further very shortly. I am an MD by training and also a little bit by practicing, and an MD PhD doing research in that building in Paris, which is on a university hospital campus and related to Collège de France. And my field is immunology and more specifically genetic diseases of the immune system. And through these diseases, although I'm not going to detail them today, this is not the scope, obviously, I will try to give you a little bit of the story about the development, but also the limitations of something which is more or less known by people, broadly speaking, that is gene therapy. So let me start with some medical aspects related to what are named inherited diseases. There are many of these genetically determined diseases that affect all tissues in the body. There are a few examples of diseases. Some of them may be known by you, like sickle cell disease, which is a severe anemia, or cystic fibrosis, myopathies, primary immunodeficiencies, my field. So altogether, we know that there are today approximately 8,000 diseases genetically determined, most with recessive or dominant inheritance. They are all rare, but globally, they affect up to 2 to 3% of the population. So this is quite significant in terms of health burden, given the high rate of mortality and morbidity generated by these conditions, that, by the way, are much simpler to study than any other diseases, because they are, if I simplify a little bit, determined by a single determinant that is a mutation in a given gene. This is, of course, oversimplification, but still it is true, which means that in this field of medicine, there have been more advances over the last 20, 30 years than in for other diseases that are much more complex, if you think, for instance, about neurodegenerative diseases. Medically speaking, the issue is treatment, obviously, and unfortunately, up to now, only few of them got a curative treatment. But this is where genome engineering comes into medicine. And of course, as you know, I'm sure, even if you are not in the field, this has developed extensively over the last 30, 40 years, with the possibility of genetic engineering, that is to manufacture, for instance, therapeutic proteins of interest by transferring corresponding gene, so DNA, into cells, and to have factories from bacteria, insects, mammals, and so on, mammal cells, to produce a given protein, like insulin for diabetic patients, growth hormone, clotting factors for hemophilia, for instance. But probably what has been the most significant advance over the last 30 years is the development of monoclonal antibodies, initially with no idea, no clue for medical application, but then it came, and today there are the drugs that are today the most frequently used in the world are monoclonal antibodies to treat chronic conditions like autoimmune diseases like polyarthritis, or inflammatory bowel disease like Crohn's disease, and also being used for the treatment of cancer with, for instance, the checkpoint inhibitors. Okay, so these are advances, despite, of course, that it does not concern or touch all aspects of medicine. But the point I'd like to discuss further today is, what about gene therapy as a genome engineering procedure? So what can we do with gene therapy? Of course, the principle is shown here. It's very simple. You take a piece of DNA or encoding material with a promoter in order to get expression into messenger RNA of the DNA, and you use a vector, as you will see, essentially a virus that has been modified in order to introduce this material into a cell, so that this genetic material will provide the cell with a given protein with a physiological function. So that's the most obvious usage. And here are the 4 different things that can be done, and some of them have been indeed implemented in the recent past. So to provide a normal copy of a gene in affected cells of someone with an inherited disease, with a monogenic disease, like the one I mentioned earlier. But it's also possible to inhibit or modify the expression of a mutated gene, for instance, to skip an exon that is mutated. You can, with the advance I will briefly discuss at the end of genome editing, consider to fix a mutation, so to restore a normal sequence of a given gene. Or eventually, you can also go to provide a synthetic gene for the implementation of a new function, and it is typically what has been achieved with the so-called CAR T cells, so chimeric antigen receptor T cells, used to treat leukemias or other malignancies with a modified receptor that can kill cancer cells. So these are the different venues of development of gene therapy. The idea, the concept came a bit more than 50 years ago. That paper probably was the very first to be published in which the idea, the concept of gene therapy was mentioned by these authors, Ted Friedman and Richard Waldblin. As you can read, that was 1972. So we are now close to 50 years later, and during a very long period of time, nothing happened actually. No clear-cut demonstration that gene therapy may work. I will try to tell you why. But if I try to give you a kind of a summary of what has been achieved, medically speaking, up to now, this is all summarized on this slide based on published results, reproducible results. As you know, this is very important because there are many hypes in many fields of medicine and especially in the context of medicine. So, but this is the chronology of publications for genetic diseases. There are 12 of them. I will not go into the details, for which gene therapy has been proven to provide a clinical benefit. So, and of course, this is the goal, not to get an improvement in a given biological marker, but to see a real improvement in the health of the patients. So in red are the immunodeficiencies I'm going to discuss a little bit more. This is my field. But you can see that there are diseases of the eye, like amaurosis, so blindness. There are other diseases of the hematopoietic system, anemias like beta thalassemia or sickle cell disease, hemophilias, and so on and so on. Different colors indicate different strategies. Ex vivo or in vitro gene therapy by using retroviral vectors, I will discuss a little bit more, used to modify cells ex vivo, usually hematopoietic stem cells, before reintroducing them into the body of the patient. And the second strategy I'm not going to develop further is in vivo gene therapy, where the gene is inserted into the genetic material of a virus named adeno-associated virus that can be used in vivo to transduce or to modify cells in the body as used for the treatment of hemophilia and other diseases. In addition to these 12 inherited diseases, as I mentioned already, since 5 to 6 years, gene therapy has been used to create this synthetic receptor to try to treat cancer, especially leukemias, and this has had a major development with limitations, nevertheless, since 5 to 6 years, but thousands of patients now have been treated by this strategy. So the field, in the context of ex vivo gene transfer, benefited very much of the knowledge about viruses, notably retroviruses. So retroviruses contain several families, including HIV, for instance, which, by the way, is used as a vector for gene therapy. They are the lentiviral vector class of retroviruses. The interest of retroviruses is that once the viral particle enters into a cell, its genetic material, which is made of RNA, is retrotranscribed into DNA associated with protein, the pro-integration complex, PIC here, that enters the nucleus more or less easily, depending on the class of viruses. And within the genome, they can integrate so that a piece of the genetic material of the virus is integrated and will be replicated each time the cell divides. And of course, this is key in the context of gene transfer because it means for a tissue like the blood, so bone marrow, where the blood cells are being developed, there is a lot of cycles of cell replication. So this is key. And then this genetic material, of course, can be transcribed, translated into proteins. So if you modify such viruses in order to make them unable to replicate, of course, we don't want the virus to replicate in vivo, and unable to produce too much of the viral material in order, again, to avoid toxicity, these viruses could be and have been engineered into vectors to treat cells. And the very first field in which this was used is the field of hematopoiesis. So just to introduce you for the those who are not biologists, the hematopoietic system, in our bone marrow, we have stem cells that are able to self-renew, and they can give rise by differentiation and division into all of the blood cells, from red cells, platelets, and all of the white cells, including lymphocytes. I will discuss a bit more. And there are many, many genetic diseases, inherited diseases, affecting the development of these cell lineages. These primary immunodeficiencies that I will discuss a bit further, like the most severe ones, the so-called SCID, which stands for severe combined immunodeficiency, But there are other diseases, like the more frequent inherited diseases of red cells, the so-called hemoglobinopathies, where hemoglobin is altered, leading to severe anemia, like in beta thalassemia and sickle cell disease. The interest for that system is that we have access, though we do not know these cells that well, but we have access to these stem cells. So if one is able to correct the genome of such a cell, the consequences may be reached into the daughter cells, the differentiated cells, and provide some benefit in the context of medicine. So what happened between 1972 and the recent past? So actually up to, say, 2000, where the first successes of gene therapy occurred. Well, we as doctors were very much relying on the development of basic research into biology. That is, as I mentioned, the discovery of these retroviruses, the understanding of their life cycle, in order to engineer them into vectors. Stem cell biology was important to understand better, although, as I said, this is not yet there in terms of full understanding, but also on a more medical aspect, the identification of genes, mutations, and pathophysiology of diseases. They were all key steps to implement prior it was possible to achieve efficient gene therapy. But it happened first, and we'll explain why, in the field of very rare diseases, but very severe, life-threatening, causing death in the absence of treatment within the first year of life. These are a series of inherited diseases causing a defect in the development of lymphocytes, mostly of the T lymphocytes. You may know that T lymphocytes are key cells in order to use to fight viruses and any kind of microbes. So those unfortunate children who are born without these T lymphocytes and sometimes other lymphocytes, they will die from infections within the first year of life. They don't have what is called adaptive immunity. So they are fatal conditions. We know that it can be treated by allogeneic hematopoietic stem cell transplantation, so to replace their bone marrow system by the system from someone who is healthy. But this procedure is associated with a number of problems. We'll not go into details, mostly the immune conflict between the donor and the recipient. So among these diseases, there are two of them, there are many more, that have been targeted for gene transfer, and I will explain why. The first one is so-called SCID-X1. X stands for the fact that the gene is located on X chromosome, so it affects boys. And those children lack T cells and NK cells. And the other disease, AGA, stands for adenosine deaminase. It's an enzyme in the purine metabolism. And when this enzyme is deficient, the lymphocytes are just absent. So these are the two diseases that have been first targeted. The question is why? How is it that these diseases were the first successful targets for gene therapy? What is the physiology and the mechanisms behind it? Well, before trying to address this question, just a word about the disease we have been treating, yes, due to the so-called SCID-X1. So we and others identified now more than 25 years ago that the gene that is defective is encoding a protein that is expressed at the surface of cells. This is the membrane of a cell. Below is the inside and outside is here. And this protein, named the common gamma chain, here in red, as you can see, can associate with other proteins at the surface of cells. And they are constituting what are called cytokine receptors. So they are receptors that get messages from soluble proteins named cytokines, like interleukin 2, 4, 7, 9, 15, and 21. And we, as well as with the help of other people, we understood in the 90s that, for instance, interleukin 7 is key to provide some proliferative and survival signals to the development of T lymphocytes within the thymus. This is where the T cells are made, for instance. So we understood the pathophysiology. And based on that, we considered that disease for gene therapy for the following reason. It is known, and this has been confirmed since, that from stem cells, there are T cell precursors that go into the thymus, in the upper part of the mediastinum, where these cells divide and differentiate and produce many, many different T cells, billions of them. And the key point is that these cells have a very long lifespan, more than 50 years. This is very much in contrast, just to give you an idea, to neutrophils, another group of white cell, where the lifespan is only 2 days, or platelet 8 days, meaning that the turnover of these cells is much, much higher. So that diseases affecting those cells will be much more difficult to cure because you will have to correct many stem cells in order to be efficient. So this is one point, and of course in the context of SCID there is a block here in the development of cells one wants to surpass by addition of a gene. But then came a story of a study we did more than 20 years ago. I like very much because this is based on a study of a single patient. Sometimes to study in detail a single patient may be very fruitful in terms of understanding biological aspects. And the point is that this was a child who had this common gamma chain deficiency, but he had T cells, and actually we could show that these T cells he had carried a wild-type gene. So actually what happened that from previous cells, like stem cells in his body, there has been a reversion of the mutation, an event that may occur, but of course very rarely, and probably only once in the life of this child. And in his blood, we could see that he had many T cells, not a normal count. He was still sick, but had only a mild disease. By analyzing the diversity of the T cell receptor of this child, it was estimated that he had 1% of the normal diversity generated from a single cell, which is quite a lot, which indicates that in the thymus, there were at least, I will not go into the detail to explain that to you, but it's obvious in terms of numbers, that there were at least 11 cycles of cell division that occurred that generated many T cells diversified by rearrangements of the T cell receptor. So these steps of extensive division was based on the expression of the common gamma chains, which tells us that the partial correction of the disease that occurred spontaneously in this child was a kind of a natural gene therapy, not fully correcting the disease, but still good enough so that the child could live without any treatment of several years. Then he got a transplantation. So this was a rationale to undergo potential gene transfer to treat that disease with the hypothesis that the cells that will be corrected among the precursors here will auto-amplify, will proliferate in the thymus in order to generate these many T cells required for a normal life. So based on this, first clinical trials were performed in 1999, so a bit more than 20 years ago, where the bone marrow cells from a child, this is a cartoon that depicted by one of the treated patients, by the way. So his bone marrow cells were taken out, harvested from his bone marrow, the most precursor cells selected, and then infected during 3 days with the virus. So it's an ex vivo experimental viral infection, but with a virus that has been modified. It's a retrovirus. This is how it looks like. Of course, this is simplified. So the idea that the virus binds to the cell, enters this precursor cell. The RNA is retrotranscribed to DNA that may, at least in dividing cells, go in contact to the chromatin, and events of recombination that can occur so that the genetic material is now part of the cell. It can be replicated each time the cell will divide, and also be transcribed into the messenger RNA, the protein transduced, expressed at the surface, and the function restored. So the first clinical trial was initiated a bit more than 20 years ago with the idea, so the theory is that this was the hematopoiesis of such patients prior to a treatment with a block in development of what is here, the T cells and another cell subset, NK cells. And the idea is to introduce the virus therapeutic, so-called quote-unquote therapeutic gene into these precursor cells in order, and this is now the presence of the gene into the cell, to restore the development of the lymphocytes. So that's the concept. So that has been done in France and then in the UK. And the results are not that bad because among the first 20 patients treated between 1999 and 2004 approximately, 17 of them are today alive disease-free, which means they are able to cope with infections. They are living normally. They are now young adults for the first patients treated. And with the correction of the T-cell immunity, I will not go into too many details. So it's not perfect, it's not bad, and this is sustained over time, which is, of course, very important. And these are the T lymphocytes in the blood. It's easy to enumerate these cells in the blood. We all have a few thousands of these cells in our blood. And prior to treatment, they didn't have any. And then, upon time, they have T-cell counts, as we say, somewhere between 1,000 to 3,000 per microliter of blood, which is within the normal range. And these cells are functioning. I will not go, of course, in many more details. Nevertheless, and this is very important to consider, these first successes were also met with toxicity. since some of the patients, 6 of them, marked here by the red arrow, develop a very severe complication, which is a T-cell leukemia that was induced by the gene transfer. So something went wrong into this procedure that needed to be analyzed in order to correct the procedure and proceed further to treat the same disease and other diseases. So in one of the 6 patients, the leukemia was fatal, whereas in the others, it could be treated efficaciously, and with preservation of the gene therapy efficacy. Of course, it led to an interruption of clinical trials, and in many efforts to assess to understand what happened, knowing that, and this is interesting to consider that in the murine models that everyone is using in biology for very good reasons, but still in that context, we never observed these leukemias, and we can discuss it if you wish. So what is the problem? The problem is that these genetic material from the virus, shown here with the transgene and the external elements, are inserting mostly within open part of the chromatin, so where they are actively transcribed genes. They are not inserting randomly into the genome. This is something we learned a little bit at posteriori. And what happened is that the enhancer element, which is contained in the viral genetic material, can induce the expression of a endogenous gene. So if this insertion occurs within a gene that is potentially an oncogene, it can be aberrantly regulated so that it is expressed constantly, so it's an oncogene transactivation. And this is the case, these are in 4 examples, there are a few more, but where insertions occurred was determined to occur within the locus of a given oncogene named LMO2, either in the promoter or introns. So this led to the transactivation of the oncogene and the leukemic process in T-cell precursors. So how to prevent this problem? Well, the solution is probably not that difficult and probably indeed works, as I will show you. It was to modify this material to remove this enhancer element, and instead of using this enhancer element to drive the expression of the transgene, to use a promoter with very little or weak enhancer activity in order not to induce this transactivation of any endogenous gene. So these vectors are named now self-inactivating for reasons I will not discuss. So such vectors were built for the very same disease, where now the enhancer element has been removed. This is the structure of the genetic material. And it was used in a new study performed between France, England, as well as the US, and with good success, not perfect in terms of control of infections, but no toxicity, no leukemia, and most 12 out of 13 of patients are alive with a good clinical efficacy in 10, and this is sustained. And this is encouraging, and even more encouraging is the application of such SIN vectors, so modified vectors, or second generation vectors, if you like, to other diseases. I will not go into the details, but just to show you that the very first 44 patients treated worldwide for different diseases using these vectors with now a follow-up which is on median of 8 years, the minimal follow-up 4.5, extending to more than 10 years. None of them has developed a leukemia, which is very encouraging, although of course we still have to be cautious about the fact that such vectors could be safely used to correct at least immunodeficiencies. Another aspect I'd like to briefly discuss derived from this study is the questions: how many precursor cells were corrected in order to obtain the development of these many T cells? And this can be determined by looking in details at the genome of modified cells by a technique of sequencing of the border, the boundary between the inserted material and the endogenous genome sequence. And surprisingly enough, there are very few. Every colored line is one given patient for the first study, and you can read that the number of different integration sites into the genome of these cells is less than 1,000, which means that less than 1,000 cells here have been sufficient to generate a normal count of T cells, and this is sustained over time. So this is quite limited, which is evidenced by the way that these few cells were able to then, because they express the protein, the common gamma chain, to amplify enough to give rise to these T cells. But the next and key question is the following. So we expect from physiological T cell development that from these T cell precursors, diverse T cells will be generated, with different spectrum of recognition of antigens from microbes in order to be efficient and to be able to fight viruses, bacteria, fungi, and so on. So is it the case? So we know that every single T cell has a distinct T cell receptor expressed at the surface required for antigen recognition, and this is generated by a process of so-called somatic recombination of these genes into the thymus, if we consider the T cells. So is this the case in treated patients? So this is the summary of the physiology. In the thymus, the cells do proliferate. This is, of course, very much simplified. At a given stage, they rearrange their receptors to generate genes with a different sequence, generating proteins expressed at the surface that are slightly different, as exemplified here, in order to get recognition of the many antigens. So is it the case in the patient? The worst scenario will be something like that, where despite a normal number of cells, there will be very restricted in terms of diversity. And of course, this would work well in terms of efficiency of immune responses. The right scenario is this one. So this is indeed what happened. These are the raw data. I will not go into details, but everything in blue indicates unique and diversity of sequences. And here are controls, adults and children, and here are patients. And you can see that the length of the blue line indicating the number of sequences that are unique are very similar to controls. So indeed, and this is a complex picture, but it summarizes everything, from a very few T cell precursors, 50 to 1,000 only that were corrected, very few cells. Those cells, once they express in the thymus this so-called common gamma gene at the surface, marked here by the yellow, if it is yellow, circle or star, I can see from here, they proliferate extensively thanks to interaction with interleukin-7. At a given stage, the rearrangement of the T cell receptor occur, leading to diversity, where different size of the different clones, but this is representing physiology, and physiology can be reproduced with only a few precursors because of this massive expansion of the transduced precursors in the time. So this is a summary of what happened. So today, how it looks in terms of clinical application. The very first step between about 20 years ago was to use this first generation of vectors. It was okay, but toxic. So we went to a second step with modified vectors, which is okay, not toxic apparently, with a follow-up of 10 years or so. But today there is even further improvement by increasing the number of modified cells and also giving a little bit of chemotherapy to patients to have more stem cells entering the niche in the bone marrow to provide more T cells, but also other cells that are important. I will not go into the details, but this is where we stand today. And the other disease I briefly mentioned at the beginning of my talk, adenosine deaminase, where all of the lymphocytes absent, has been treated by the same technology. I will not go into too many details. In a first wave of treatment, of trials performed in Italy, US, and UK, most of the patients have been efficiently treated. No toxicity, surprisingly, compared to the other disease, for reasons I have no time to discuss. And importantly, one of this drug product, because this retroviral vector, because it works, not in all patients, but in many of them, without toxicity, is now on the market, is an approved drug product, which is the first real success in terms of marketing of a gene therapy product. And more recently, new vectors based on lentiviral vectors from HIV that are much more efficient are being used successfully for that disease too. So at this stage, the conclusions are as follow. As well, with these many steps, proof of principle of efficacy has been provided in a very favorable setting in terms of gene therapy, because the few cells corrected can expand and then are long-lived, unlike neutrophils, as I told you earlier. The third benefit is sustained, and this is based on this so-called auto-amplification. The safety was an issue, but hopefully has been solved by removing this enhancer element from the vectors. Some chemotherapy is useful. And because of that, of course, now there is extension to treatment of other diseases. I will mention only one, which is the so-called Wiskott-Aldrich Syndrome. It's another X-linked disease where there is an immunodeficiency, which is more complex, but also a problem with platelets. So there are bleedings, as you can see on this unfortunate boy. These patients also develop autoimmunity. And the protein that is deficient, the so-called Wiskott-Aldrich Syndrome protein, is related to the organization of the actin cytoskeleton in blood cells. So to give you a short summary, a vector has been built where the cDNA, so the coding material from the gene, has been inserted into a lentiviral vector under the control of the own promoter of this gene. And this vector has been used to transduce cells ex vivo, the same technology, whereas in between patients got some chemotherapy. And we performed a study with our colleagues in London, for instance, where in 7 patients, a clinical benefit has been clearly observed. There is a score of the disease, 5 being the worst, 0 is normal. So the score went down from 4.7 to 0.3. With the follow-up you can read. And without toxicity. It's not perfect. For the correction of the platelet disorder, for instance, it's not as good as for the one of lymphocytes, because for the reason I mentioned earlier, you need many more stem cells to be corrected to provide every day the number of platelets required for full functioning of the coagulation system. But it's good enough so that the patients are doing well. So based on that, there are further development for other immunodeficiencies, other severe combined immunodeficiency, other T-cell immunodeficiencies. There are more than 50 to treat, so it's a huge development in terms of resources that is being used. And then next step that becomes harder is to treat diseases for which gene transfer does not confer this auto-amplification I discussed in detail, plus to combine it to treat to target cells with a much shorter lifespan like neutrophils. There are also genetic disorders of neutrophils. This requires a very robust and efficient transduction of stem cells. We are almost there, but not fully. We still have to make advances in terms of technology. And then comes the issue of treatment of thalassemia and sickle cell disease. This is even harder because then you need to have a restricted expression of the transgene, hemoglobin in that case, in the erythropoietic lineage. There are some advances on this, also for other diseases I have no time to discuss. But these are ongoing clinical trials that are providing some hope for the future. To finish with, in the last minute, I'd like to discuss where do we stand and where we are, which is actually not something very sophisticated. What we are doing these days is something like that. Let's consider a stem cell of the hematopoietic system. And we are and there is, for simplification, only one allele is shown from a given gene. And you have in the red dot indicates a mutation. So the gene doesn't work properly, like ADA or common gamma chain or others, or Wiskott-Aldrich Syndrome protein gene, for instance, so that there is a disease. So what we do, we are manufacturing a virus, a retroviral virus, to become a vector, to introduce into the cell, into the genome somewhere, but this is not controlled. And as you saw, it was a problem in the past. So somewhere in the genome, there is kind of semi-random integration of the normal copy of the gene in order to get the normal product. This is okay, but far from being perfect because we are not respecting physiology. This gene is not under its physiological control, as it should be. For some genes, this does not matter much, like for common gamma chain or ADA, but for others that are under very strict regulation, it is not going to work and it could be harmful. So can we do better? Well, so there is, in this context, as I said, no physiological control of it. Can we do better? And this is where comes the genome editing strategy that I think everyone, not only biologists, have heard about. That is now today we can use modified nucleases, engineered nucleases that can cut the DNA at a very, very precise place. And then we can do cut and paste in order to introduce some pieces of material, like a small one here in green or a bigger one at a very given place into the genome. So perhaps to fix a mutation and get physiological control of the modified gene. Something else one can do, which is of interest in some cases, is to destroy a gene. Maybe to destroy the mutated gene, for instance, by inducing a cut without providing any matrix for recombination, so that the cut will be repaired, but will be repaired not in a clean way, so that mutations are introduced and the gene is disrupted. That is pretty easy to do, actually. And probably the first clinical application will come for that strategy, but it is limited to inactivation of a gene. There are not so many instances where we want to inactivate a gene. Here is the more demanding strategy of homology-directed repair, providing a template and having homologous recombination, so a clean repair without generating any mutations. As you know today, there are many tools to do that for performing it. There are the first two that were developed over the last 10, 20 years, or engineered protein-guided nucleases, where so there is an enzyme that will cut the DNA, and the code for recognition comes from another protein, so-called the zinc finger nucleases or the TAL effector nucleases, that have been engineered to cut at certain places, but they are not easy to manufacture because not all of the places into the genome can be targeted. The code of recognition is quite complex and they are so cumbersome to design. More interesting, perhaps, although there are some advantage with this system too, is the very famous system of the CRISPR-Cas9 system, where now the guiding of the enzyme to the right place comes with RNA, which is complementary of the DNA sequence you want to target. And as you know, this is these days used in every single biology laboratory in the world to modify cells, to modify animals. So the question whether it can be used in humans to treat patients with genetic disease, for instance. So the idea here, let's take the same scheme I showed you about on that left-handed part, that this is gene addition. Now this is potentially gene repair of the very same mutation. Then you have to introduce, again, perhaps with a vector, but other means could be used too, the DNA templates for recombination. And then, of course, a second step can be done together, is to provide the cell with the engineered nuclease, say the CRISPR-Cas9, for instance, in order to target the DNA close to the mutation to get homologous recombination and repair of the mutation. Of course, that will be ideal. This is the theory, but it's not that simple. It requires the so-called homologous recombination machinery. It's the machinery to repair DNA, and that has to work in a very clean way in order not to generate mutations, of course. And this works only in cells that are dividing, not so in stem cells. So it's a major bottleneck for application, at least in the field of the hematopoietic system. Maybe this system may be good enough to correct diseases like severe combined immunodeficiencies, since I showed you that only a few cells are enough. If you correct, say, a few hundred cells, this may be sufficient. So the technology may be applicable for that diseases. But for other diseases like thalassemia or other diseases, this may be much more difficult for the limitation I just mentioned about the availability of the homologous recombination. And then there is also the issue of off-targets, that is that the system may from time to time generate cuts outside of the region you want to target, and that may be a problem too. So, which means that today we are not there yet. And this may require many more years. We do not know. It's impossible to predict about when this technology will be usable in patients in a real clinical world. So another strategy potentially is base editing, where you modify the base of the DNA without inducing any cuts, and that, of course, will be very much of interest, although, again, the off-targets issue is there. So a summary of what happened over the last 20 years is it started about 20 years ago with the usage of gamma retroviruses to target precursor cells, and that was enough to correct SCID, but with some toxicity. The addition of chemotherapy, as proposed 2 years later, increases the capacity of transducing hematopoietic stem cells. Of course, this is of interest on the long term. The generation of these safe vectors has improved safety, and this is since now a bit more than 10 years. Usage of now lentiviral vectors, another class of retroviruses, has increased the capacity of transduction, so to correct more cells, and this is important for some diseases. Then came for other diseases the capacity of generating more supraphysiological level of expression of the transgene. This is interesting for some enzymatic diseases, for instance. And also addition of further regulatory material is used, like so-called locus control region addition, to get lineage-restricted transgene expression, like hemoglobin in red cells. This has now been used efficiently. So these have been the advances over the last 20 years. And maybe for the future will come genome editing with engineered nucleases for inactivation of genes. Again, this is probably not too difficult, or replacement. and maybe eventually base editing, we'll see. So to finish with, what I told you was a long journey, actually, full of serendipitous findings, like the discovery that a few cells were enough to correct some diseases. So proof of principle of efficacy and safety eventually provided, but this was lengthy and very painful, I can tell you. But now we may also consider, and this is interesting on a biological point of view, that gene therapy can be used as a surrogate to study stem cells and lymphocyte dynamics because we have a gene marking of cells. So we can trace these cells in the blood of these individuals and do study about how they develop, they survive, they differentiate, and so on. And the next steps may be related to the development of these engineered nucleases I mentioned. We'll see in the future. Finally, just to mention some names of people involved in our part of this work. Mostly these 4 people, the names shown in blue: Marina Cavazzana, Salima Hassenbey Abina, Geneviève de Saint-Basile, and Jean Lim Santo, who did all of the preclinical and clinical work with the help of many, many collaborators worldwide. Thank you very much.